including BBSRC, David Phillips Research Award, the Commonwealth PhD Studentship, working in the area of cancer, diabetes, and how interrelated morbidities, and where he will be talking about how these interrelations can go on, it can give some inputs in the COVID-19 research. And during the session, if the participants have any kind of questions, hello?
Hello?
Yeah, sorry for some disturbance.
Yeah, that's good.
Yeah. During the session, if the participants have any kind of questions, please use the chat option to type in your questions, which we will try to answer by the end of the session. With this, Professor Venkat, yeah, please. Sir.
Okay, thank you very much, Dr. Somasundaram, and also Sudhakar and Loganathan for giving this opportunity to present our work in this conference. So just as title suggests, I'm going to discuss with you about the treatments we are trying to develop for diabetes, cancer, and also how we can relate those to COVID-19, which is kind of an important issue in the present time. So I'm going to talk, it gives an overview of what I'm going to talk is shown in this slide. I'm going to talk about type 2 diabetes. One of the important drug target for this disease is called GLP-1 receptor. And I'm going to talk on related to how it gets transported to the plasma membrane and how it gets internalized, and some small molecular agonist for this GLP-1 receptor. Because it's an important drug target, we want to know various aspects of this receptor, and thereby it can be useful for making better drugs for type 2 diabetes. And then I'm going to talk about lytic peptide targeting of breast cancer. We developed a ligand fused with a lytic peptide, which specifically targets breast cancer. We also studied for prostate cancer, but due to the limitation of time, today I'm going to concentrate on how we can use this peptide for targeting the breast cancer. Finally, I'm going to briefly discuss with sepsis, which we have been working to use we work on a small GTP binding protein called OF6, which is known to regulate sepsis affected by the bacterial infections. And because recently studies shown that COVID-19 can also cause organ failure, it's a similar to that of by sepsis. We're wondering whether we can use the inhibitors we already using for bacterial-induced sepsis for studying COVID-19 associated organ failure through vascular leakage. So let's look into the type 2 diabetes. One of the important hormone that regulates insulin secretion is known as GLP-1, which is a glucagon-like peptide 1, which regulates many physiological functions, which are all illustrated here. The main function is it controls insulin secretion in a glucose-dependent manner. This made it a very important drug because most of the conventional diabetic drugs works glucose-independent manner to regulate insulin secretion, and therefore you have to measure glucose levels in order to take the required dose to control insulin secretion, such as metformin and other conventional type 2 diabetic drugs. Whereas GLP-1, the advantage is that you don't have to measure glucose levels. You can take one specific drug dose, and then it can control insulin secretion based on the blood glucose levels. And therefore it became very popular drug in recent years mostly in Western countries. If you look into the endogenous GLP-1 hormone, it has only 1.5 minute half-life period. This is because it exists in 2 isoforms, they're shown here, but it's broken down by a dipeptidase 4 into a inorganic metabolites, inactive metabolites, which no longer binds to the receptor on in their own, and therefore they cannot induce insulin secretion. So to overcome this problem, many companies developed peptidase resistant GLP-1 mimetics. For example, if you take a liraglutide, which is a resistant or stable form of GLP-1, which it has an half-life of about 12 hours. Therefore, you need to take on regular basis to control type 2 diabetes. Whereas exenatide, which is a kind of a isolated originally from lizard saliva, and it has an half-life period of about 4 hours, and therefore you have to take twice a day to control as a medication for diabetes. However, these drugs have been modified in such a way that you can improve half-life to almost 96 hours, about 4 days, by modifying these compounds. One of them is called dulaglutide, where you can attach 2 liraglutides to an IgG molecule, or another one is called exenatide, which can be formulated in such a way that it can be increased its half-life by to 4 days. One of the problem with these drugs is that you have to take them as an injection. Therefore, if you have to take every day, then it's very inconvenient. Therefore, a lot of companies are working to make various forms so that you need to take only once in a week or maybe once in a month in probably near future. So as you can see, type 2 diabetes is the main common form of diabetes. And in the world, maybe 200 to 300 people are suffering from this disease. In fact, there may be many more, but we don't know because many people are not aware that they have the type 2 diabetes. And therefore, because of the huge population and they need a regular basis, the treatment, there are many pharmaceutical companies working in developing various forms of GLP-1 mimetics, which mainly works on GLP-1 receptor. As you can see, there is AstraZeneca, Novo Nordisk, GSK, Eli Lilly, Sanofi, and Roche, and so many other big pharmaceuticals are working because it's an important area to develop drugs. So, as I said, GLP-1 mainly works or functions by acting through GLP-1 receptor. So, what is GLP-1 receptor? Obviously, it's an important target for type 2 diabetes, and it is a G protein coupled receptor. Upon agonist binding, that means GLP binding, a GLP-1 receptor couples to GS pathway to increase cyclic AMP production. And also, it can also couples to the GQ pathway to increase intracellular calcium and accumulation as well as EC phosphorylation. Once receptor gets activated, it gets internalized to dampen the biological response or to resensitize the receptor through recycling. So what I'm going to talk today is about, start with looking at the importance of the N-terminus of the receptor for surface expression of GLP-1 receptor. Because these are cell surface receptors, they have to be functional at the localized at the plasma membrane to be functionally active. So GLP-1 receptor is a G protein coupled receptor, as I already mentioned. It has a signaling peptide that needs to be cleaved off for the protein for the receptor to target to the membrane. So what we have done is we assess the importance of cleaving of the signaling peptide by overexpressing by expressing this receptor in HEC cells and studying the localization at the plasma membrane. As you can see here, there are different mutations we made. We take a wild type, attach it GFP so that we can see the receptor under confocal microscopy or fluorescence microscope. And then we also added a epitope tag so that we can add antibody to recognize the localization of the receptor within the cell. So we have the wild type Here we have deleted the signal peptide, and this mutant where we replace signal peptide using a vascular signal peptide, which no longer cleaves off from the receptor, and or we also mutated a residue called alanine at position 21, which makes cleaving of signal peptide difficult. So then we expressed these cells in hex cells and studied the localization. As you can see, there are two bands here. That is the mature form, which mainly localized to the plasma membrane, and this is the immature form, mainly it's intracellular. So this is what we are looking. This is the functionally active receptor. So when you take the wild type, you can see most of it, some of it is at the plasma membrane in active conformation. Whereas if you remove the signal peptide, it also goes to the membrane because signal peptide cleavage is required for the receptor to target to the membrane. And then when we replaced signal peptide with a different signal peptide, which is here, it's a viral signal peptide, which we know that it can't cleave off from the receptor due to the different substrate sequence differences. As you can see, when the signal peptide is no longer cleaved off, then you don't see the expression of the receptor at the cell surface. Similarly, if you mutate the receptor so that it cannot cleaves off the signal peptide, then also you don't see receptor being localizing to the plasma membrane. So we have used two different antibodies to show the same data. This is the anti-VSVG antibody, which binds here, and this is GFP antibody, which binds intracellular one. So based on that, we show that signal peptide cleavage is required for the protein receptor targeting to the membrane, which we also confirm by flow cytometry. As you can see, by flow, you use antibodies without permeabilizing cells, so that antibody binds the extracellular portion of the receptor, and thereby you can measure how much is outside the cell. As you can see, wild type, and this is the signal peptide deleted construct, and these are the mutants which fail to cleave off the signal peptide, and therefore they are mainly localized inside the cell. Because the receptor has to be at the plasma membrane in order to be functionally active, therefore we can see that the receptor at the membrane, you have the active cyclic AMP production. Similarly, so it kind of reflects what we are seeing the localization. Receptor at the membrane, plasma membrane is active, and therefore it produces cyclic AMP product response. Whereas the receptor which is inside the cell is no longer active because it doesn't able to, it no longer get access to the ligand which present outside the cell, and therefore it no longer produce cyclic AMP. Similarly, we also established the localization by immunofluorescence. Here, green color indicates GFP, and red color shows the antibody binding through VSVG tag. So the VSVG tag is only present outside the cell of the receptor, and therefore if the binding, if you see any red color, that means it is localized at the cell surface. So this is wild type. You can see the receptor is at the cell surface because of the red color, and here also you can see signal peptide cleaved one also at the surface. Whereas if you either modify the signal peptide or mutate, it's no longer localized to the plasma membrane. That's why there's no red color staining here. So if you look into overall this one, it suggests that the cleavage of the signal peptide is required for GLP-1 receptor cell surface expression. Then we looked into the region called hydrophobic region adjacent to signal peptide requirement for the receptor cell surface expression. Again, we made a series of deletions. As you can see, the number indicates the number of amino acids being deleted at the end terminus. This is the wild type. We have removed 24 amino acids, 30, 35, 40, and 31 to 40 amino acids at the end terminus, and studied their effect on the localization. When we remove either 35, 40, or the region between 31 to 40, we see there's no localization to the plasma membrane of the receptor. Because these mutants are not at this membrane, at the cell surface, they're no longer active. in producing cyclic AMP response, or this cell surface expression or the cyclic AMP production, which also come from through the immunofluorescence. You can see there's no red color in these deletion constructs, suggesting that you need this region that is 31 to 40 amino acids for the cell surface expression of the receptor. So that is the region we consider as hydrophobic region after signal peptide, which is essential for the receptor cell surface expression. Very often receptors do undergo glycosylation, and glycosylation is also important part for the receptors to target to their own localization. In this case, it's plasma membrane. So very often deglycosylated receptors run faster than the normal N-glycosylated receptors in Western blotting. So as you can see here, this is the wild type. When you treat with the tunicamycin, which is an inhibitor for glycosylation and glycosylation, and you see there's no glycosylation of the receptor, therefore it runs faster than this one. So it appears as a low molecular weight receptor. Whereas if you delete N-terminus 145 amino acids, which contains all the required glycosylation sites, then it no longer goes to undergoes N-glycosylation. Therefore, there's no shift in the mobility because there's no glycosylation to start with, and therefore it cannot be inhibited further by tunicamycin. Similarly, if you mutate 3 amino acids which we know are required for glycosylation, that is aspargine 63, 82, and 115 to leucine, then no longer gets glycosylated, and therefore still you don't see the effect of tunicamycin treatment on the mobility of this, indicating these are the sites required for the N-glycosylation. We also come from through using an enzyme treatment. There are two types of enzymes called EndoH and PGN-ASF, both deglycosylate N-glycosylation proteins in this case, but they use a different substrates. So the N-glycosylation of GLP-1 receptor is more prone for PGN-ASF, and as you can see, it's affected the mobility, whereas this enzyme is not, indicating the N-glycosylation of the receptor is more sensitive for this enzyme. Again, when we treat with the tunicamycin or the mutants which are defective in N-glycosylation, they no longer express at the cell surface, and therefore they are inactive in producing cyclic AMP, indicating that N-linked glycosylation is also essential for the receptor's cell surface expression. And also there are mutants which previously shown to be required for the activity of the receptor. We wonder if some of them are actually indirectly affecting the activity of receptor by mainly preventing the receptor translocating to the membrane. Therefore, what we have done is we recreated those mutations and studied the localization. That is, we converted glutamic acid into lysine, which is at 34 position. And similarly, we kind of mutated this amino acid at 39th position, and this one at 69, and again, and the tyrosine at 88. And then we studied the localization. Even though it is E34K mutant is defective in ligand binding, we can still see it's localizing to the membrane. The top band indicates membrane localization. Whereas mutating the other 3 amino acids, which previously shown to be required for the activity of the receptor, are no longer localized to the membrane, indicating that the receptor is inactive because it's not localizing to the plasma membrane when these amino acids are mutated. So we confirm that by cell surface expression analysis. As you can see, these 3 mutations will prevent the receptor targeting to the plasma membrane, and thereby preventing binding to ligand and cause the activation of producing cyclic AMP. Again, we confirm by immunofluorescence as well. So the localization is again indicates that it's very important for the receptor activity. So we looked whether the non-glycosylation mutations, whether they are kind of affect the receptor targeting to the plasma membrane by inhibiting N-glycosylation. As you can see, when you make signal peptide mutations, if you remove the replace the signal peptide with a vascular signal peptide or mutate the signal peptide cleaving, then there's no N-glycosylation, indicating that yes, these mutations affecting N-glycosylation and thereby targeting to the plasma membrane. And similarly, when you make a series of deletions at the N-terminus, and we know that this is required for, this region is required for the membrane, plasma membrane targeting of the receptor, and when you make this mutation, then there is a reduction in the glycosylation, indicating this region also required for the glycosylation. Similarly, when we mutated, these three amino acids also somehow affected receptor targeting to the membrane by inhibiting N-glycosylation. So based on this data I've shown so far, we kind of proposed a model. According to this model, GLP-1 receptor, when it's synthesized in the signal in endoplasmic reticulum, this is outside of the ER, this is inside of the ER. And we proposed that maybe signal peptide somehow masking the N-glycosylation sites. When the signal peptide gets cleaved off, then which release the exposure, which causes the exposure of the N-glycosylation sites, and then gets when they get glycosylated, then along with the hydrophobic region after signal peptide, together play an important role in processing of the receptor in the endoplasmic reticulum, as well as in Golgi and to translocation further to the plasma membrane. Then we also looked into the, I want to emphasize one bit. As I mentioned before, GLP-1 receptor is important, internalization is important for insulin secretion. So we wondered what pathway receptor uses for internalization after being exposed to the ligand. So we first looked, because as you may remember, GLP-1 receptor kind of initiate or activates two different pathways. One is GS coupled pathway, another one is GQ coupled pathway. So what we have done is we've taken different inhibitors, or for example, this is the activator of adenyl cyclase, or the adenyl cyclase inhibitor, which kind of in the GS pathway, and also PK inhibitor, which is a protein kinase A inhibitor called H89. And we used this one along with a plasmid mutant, which dominantly inhibits, dominant negative mutant, which inhibits GQ pathway, and studied their effect on cell surface expression of the receptor by ELISA as well as by immunofluorescence. As you can see, only when we inhibit the GQ pathway, we saw the inhibition of the internalization. Whereas same thing here also. So this is the receptor at the cell surface. When you treat with cells with GLP-1, you can see this dotty appearance inside the cell, which indicates internalization of the receptor. And that internalization fails when we treat cells or co-transfected with the GQ inhibitor, indicating that GQ pathway is required. So we went and looked, because there is a classical GQ pathway, which result in, we looked whether that is involved in GLP-1 receptor. So the pathway has been shown, is shown here. So GLP-1, when it binds to GLP-1 receptor, which result in activation of the GQ pathway. GQ, when it gets activated, it activates phospholipase C, which then hydrolyzes PIP2, which is a phosphatidylinositol 4,5-bisphosphate into IP3, which is inositol 1,4,5-trisphosphate, and DAG, diacylglycerol. So just for simplicity, I just say IP3, which binds to IP3 receptor, which result in release of on the endoplasmic reticulum, which result in release of calcium from ER into the cytoplasm, and increase in cytoplasmic calcium concentration leads to PKZ activation, which then cause phosphorylation of work through RAP pathway, which may result in GLP-1 receptor internalization. So we hypothesized this, and we studied that by using various chemical inhibitors, and studied whether they inhibit internalization in a dose-dependent manner. For example, we looked here, there's an inhibitor called U73122, which inhibits PLC. So when we add this one, then we no longer see internalization of the receptor, and which we measured by LSI here. So the inhibition of the internalization here, and similarly, you can see inhibition of the internalization of the receptor by immunofluorescence. So we then used the PBP10, which is a small peptide which binds PIP2 and thereby prevent the availability of PIP2 to PLC for generating IP3. Again, PBP10 also inhibited the internalization in a dose-dependent manner in both ELISA as well as in the in immunofluorescence, you can see here. Then when we treated with the IP3 receptor inhibitor, or we kind of chelated calcium, cytosolic calcium, or inhibited with PKC, we have seen a dose-dependent inhibition of the internalization. Similarly, with ERK phosphorylation also caused this, indicating that GLP-1 receptor is downstream of this pathway. So we also looked into the effect on the ERK phosphorylation. As you can see, PBP10, which is a kind of a sponge up the PIP2 levels, and similarly PLC inhibitor, and IP3 inhibitor, calcium chelator, and PKC inhibitors, and ERK phosphorylation, all inhibited ERK phosphorylation. Just to make sure that they are specific, we also used controls, which are the inactive forms of various inhibitor we used. For example, U73343 is an inactive form of U73122. So when used inactive form of the inhibitors, we don't see any effect on the internalization or ERK phosphorylation, indicating this the inhibitors are specifically acting on the internalization. So this is the way how we analyzed the importance of the particular pathway for GLP-1 receptor internalization. As I mentioned earlier, GLP-1 receptor internalization is very important for insulin secretion. Therefore, it's very critical for us to analyze the pathway involved in it, so that we can able to improve the ligands, so that we can increase insulin secretion for a longer period. So we also looked into the importance of the C-terminal domain of the GLP-1 receptor, because it is known that C-terminus of GLP-1 receptor is required for the internalization process, because it binds to GS or GQ pathways and thereby regulate the internalization. However, we also want to know whether that also affect the cell surface expression. Therefore, we made a series of deletions. N450 means a GLP-1 receptor is about 463 amino acids in size. So N450 means there is a 450 amino acids left in this deletion mutant. That means we deleted 13 amino acids. Here we deleted 20 amino acids and 23 amino acids and 33 and 53 amino acids, and studied the at the C-terminus, and studied the effect of the deletion on the cell surface expression of the receptor. As you can see, when we deleted lost 53 amino acids, we see loss of the cell surface expression. We then made a series of internal deletion, and according to this deletion, if I summarize this bit, this is important for the cell surface expression. So the amino acids between 411 to 418 is required for the cell surface targeting of GLP-1 receptor, which we also come from. As you can see here, wild type, you can see red color, whereas when you delete 411 to 418, it's no longer shows red color, indicating that it's important for cell surface targeting of the receptor. So again, we look for the importance for N-glycosylation, and we confirm that like again it's affected by 410, or we can, when we delete, we see the effect on glycosylation by this deletion mutant, again indicating that it's somehow affecting the receptor targeting by affecting the glycosylation. So then we looked into the activity. As you can see, when we deleted the loss up to 410, we saw the on the cyclic MP production, and similarly when we made this cyclic MP is affected by this, it's because it's no longer localized to the plasma membrane. Then we also looked the effect of the various deletion at the C-terminal domains on the internalization of the receptors. This time we choose only mutants which we already know that express at the cell surface, and look for the effect on the internalization. When you stimulated cells with ligand and measure the receptor after stimulation to study how much internalization took place. As you can see in this one, we see a lot more decrease in the N433. That means when you delete last 33 amino acids, we are seeing the effect of it on the internalization. When we made a series of internal deletion, we could kind of reduce that further to show that 431 to 450 amino acids are required for the receptor internalization. Again, we come from by immunofluorescence. This is the unstimulated one, various mutants and wild type. These are stimulated with the GLP-1 or liraglutide. The red indicates the internalization, the surface expression here, or internalization, because when it gets internalized, you can see in the red color in a punctate form, and that is affected by deleting the amino acid between 430 to 450 amino acids. We also looked into the importance of earth phosphorylation by using the same mutants. Again, this confirms that the mutants that are defective in internalization also defect in earth phosphorylation. So if I summarize this part of the talk, as you can see, we kind of systematically analyze the importance of various parts of C-terminus for different aspects of the receptor. 411 to 418 amino acids of the receptor are required for cell surface expression, whereas 419 to 430 is required for the GS coupling, whereas 431 to 450 is required for internalization. As you may remember, I mentioned one of the problem with this GLP-1 receptor agonist is that you have to take them in the form of injection. That too, if you have to take regularly, either once or twice a day, it is very inconvenient. Therefore, there's a lot of interest in developing drugs that are orally active. So the company has developed few things, several small molecules that are that can be taken orally. So what we have done is here we taken two examples. One is called compound 2, another one is called compound B. Use these two, studied how they regulate receptor, how do they activate GLP-1 receptor with when compared to that of GLP-1 endogenous ligand GLP-1. So when we compare, though we need a lot more higher concentration of the agonist, we can get a cyclic activation of the receptor to the same extent as a GLP-1. But surprisingly, these small molecules don't induce calcium accumulation, indicating that they don't cause or they won't couple to the or they won't activate the GLP-1 receptor to couple to the GQ pathway. Similarly, because ERK phosphorylation is also downstream of GQ pathway, these small molecules fail to activate, cause ERK phosphorylation as well. Since the GQ pathway is required for the receptor internalization, again, we haven't seen the effect on internalization by these compounds, compound 2 and B, cannot induce or did not induce the internalization. We then looked whether the binding site is same for native ligand as well as for these small molecules by using an antagonist, which is shown here as exenatide 9-339, which we know that it's competitively inhibits a GLP-1 binding to the receptor. So as expected, it reduced the cyclic heme production by competitively inhibiting the GLP-1 binding, but it has no effect on small molecule binding binding to the receptor. Similarly, when we use the cell surface expression as a readout, this agonist could inhibit GLP-1 receptor induced internalization, whereas we know that we couldn't do that with small molecules because we know that they don't induce cell surface internalization. So we also made 2 mutants, which we know one is required for GLP-1 binding, one is required for coupling to GS, and studied the effect of these mutants on activation of the receptor by either GLP-1 or compound 2 or compound B. Because V36A known to inhibit GLP-1 binding, when we made this mutation, the receptor no longer binds to the ligand, and therefore we don't see any production of cyclic AMP. Whereas here we can see, because it's decouples to the GS, also we see a reduction in GLP-1 induced cyclic AMP production. Whereas compound 2, we know it binds at a different site. It has, that binding is not affected by V36A mutation. But nevertheless, even though it binds at different site, it does cause coupling to the GS and thereby increase in cyclic AMP production. Therefore, when we make this mutation, we see a reduction in cyclic AMP production. Similarly, compound B also just mimicked compound 2. And caused a similar kind of the effect. And therefore, what it tells is that compound 2 and B bind at a site different from that of the native ligand, but they couple to same GS pathway. So it just summarized here, compound 2 and B mainly activates the GS pathway, which results in increase in cyclic AMP that is required for insulin secretion. In addition, they could also, I haven't shown the data, but I have the data to show that it can also inhibit the GQ pathway activated by the GLP-1, and thereby it can reduce or inhibit internalization induced by, mediated by GLP-1. Therefore, what we are trying to say is that these compounds probably better than GLP-1 for several reasons. Compound 2 and D can be taken orally, and also probably cost-wise also cheaper, because the GLP-1 is a peptide. It is very expensive. I think it costs about 2,000 to 3,000 pounds per year to take this treatment, whereas compound 2 and B, if they come into market, probably they cost you a fraction of that. So second part I'm going to talk about is about the targeting of the cancer. So here, very often major problem with the cancer is the treatment, because when we use a chemotherapy, we tend to chemotherapy, very often the drugs we use for chemotherapy cannot differentiate between actively multiplying normal cells and cancer cells. And therefore you see a lot of side effects when you use a chemotherapy for many types of cancers. And therefore there's a lot of interest and demand for developing a therapeutic targeting of cancer. So many groups are working to target using different approaches. One is either you can use antibody. Oh, sorry, is it using antibody as well as any other ones? What we have chosen is we chosen a peptide that specifically binds to there is a receptor called IL-13, the interleukin 13 receptor, which overexpress mainly in several cancers, including breast cancer. So we thought, can we make a hybrid peptide which contains a small ligand that binds to alpha-2, IL-13 receptor alpha-2, and the lytic peptide that known to lyse the mammalian normal cells. So what we have done is we kind of created a hybrid peptide and tested whether it specifically target breast cancer. So just a brief introduction for those who are not familiar with this part of the subject. So interleukin-13 cytokine plays an important role in allergy and atopic diseases. It has two receptors. One is called low affinity receptor IL-13 receptor alpha-1, and also it has an high affinity monomeric receptor alpha-2. Alpha-2 receptor is kind of decoy receptor because it has no functional domain until recently, but recently people did identify some functions. But for a common purpose, we can consider this as a decoy receptor that is highly expressed in many cancers. And therefore it can be used as a drug target. So to target cancers, we developed a lytic peptide by fusing a PEP1 to a 421, and as you can see, to kind of to target cancer cells that overexpressing IL-13 receptor alpha-2. So if you look into the PEP1 421 peptide, it composed of an IL-13 alpha-2 binding moiety, which is called PEP1, and also a cell membrane disrupting peptide 421. 421 cannot lyse the cells unless it reaches a particular concentration, and that can be achieved by targeting, by binding of PEP1 to the receptor that mainly expressed in cancer cells. So we tested whether this PEP1 421 specifically kills cancer cells in 2D as well as 3D cultures. So before doing that, we want to measure what is the expression levels of alpha-2 in breast cancer. As you can see, the expression is quite high. This is a log scale. So you can see the expression is quite high in cancer. If you subdivide, the expression is even higher in triple negative breast cancer when compared with non-triple negative breast cancer. This is by RT-PCR. As you can see here, we have done by immunochemistry. The stronger the signal, that indicates the more expression of the receptor. And as you can see, this is isotype control antibody. Or here we used an antibody specific for IL-13 receptor alpha to indicate a very strong signal, indicating the expression is high in triple negative breast cancer. We choose the cell line models. We choose MCF10A, which is a non-tumor cell line, MCF7, which is a breast cancer cell line, but it's a non-triple negative breast cancer homolog. And here we used a 231 as well as its derivative LM2. Both are triple negative breast cancers. As expected, you can see the expression alpha-2 expression is high. This is done by Western blotting. It's more in triple negative breast cancer cells. And also we can see they have a relatively high expression of the receptor at the cell surface. This is done by...
Sorry to interrupt.
Hello, yeah?
We have only 10 more minutes, sir.
Okay, yeah,
Please.
sure, I'll go quickly.
Yeah.
So when we express this in COS7 cells, which are negative for alpha-2, and studied, as you can see, cell viability is reduced when you treat it with hybrid peptide, or also you see an increase in cytotoxicity. And also it is specific because it only specifically kills cells that are expressing alpha-2, but not luteinizing hormone receptor. And the way it kills is the receptor gets target attached by this ligand, thereby brings this lytic peptide into the close proximity of the plasma membrane, which result in disruption of the plasma membrane, thereby result in cell lysis. So we looked into cell lines. Again, you can see here, these are IL-13 positives. You see more cytotoxicity in those cells. And it's mainly through necrosis. And MCF7, because it has low expression, we looked whether we can increase the expression by using epigenetic drugs, which are trichostatin A and azacitidins, which are known to increase expression of certain types of genes. As you can see, in MCF7, we saw an upregulation of IL-13 receptor alpha-2. And because of that, we see an increase in the sensitivity of MCF7 cells to this peptide when we treated these cells with epigenetic drugs, TSA or azacitidine. And also we looked into the 3D culture. Again, you can see expression is very high in 3D spheroids of the same cells, and also 231 spheroids and LM2 are more sensitive to this peptide. We also done by immunocell imaging. Red color indicates cell lysis. So when we treated with PEP421, only 231 and LM2, you see the red color indicating cell lysis, whereas MCF7 and non-tumor MCF10A are negative. Again, if you take spheroids and treat with this epigenetic drugs, we see only increase in alpha-2 expression in MCF7 spheroids that treated with TSAR 5-azacitidine, and thereby we see an increase in the sensitivity of MCF spheroids for the lytic peptide. So this is kind of more relevant to the COVID-19 work. Very often, we've been working for a while to study the OF6 small GTP binding protein requirement for sepsis treatment. If you look into so what is sepsis? Sepsis is very often originate in the lungs by bacteria, and they're very often they're multi-drug resistance. When they enter into the bloodstream, which cause leaking of blood vessels, thereby organ dysfunction and death. There are about more than 5 million deaths annually due to sepsis, which also cause other complications such as septic shock. If you look into the intracellular pathway, the binding of bacterial component to TLR4, toll-like receptor 4, result in activation of two different pathways. One is mediated by NF-kappa B, which result in inflammatory cytokine production, which required for fighting against the infection. And then the other pathway is through OF6 ADP ribosylation factor 6, which result in cytoskeletal reorganization, thereby creates gaps between cells. So we developed a novel assay, which shown in cartoon by here. You have the biotinylated gelatin, monolayer of endothelial cells, and then you have a FITC labeled streptavidin. In unstimulated cells, it cannot pass through the gaps to bind biotin. Whereas when you stimulate cells with so that they become permeable, then the streptavidin pass through between cells and thereby binds biotin at the bottom of the well. So we have done that by using LPS. We stimulated epithelial cells, shown here, this is a cell line, and then studied the streptavidin pass through the gaps. The green indicates more permeability. When we increase the dose, we see an increase in the green color, indicating there's an increase of the permeability. So when we treated with various OF6 inhibitor, OF6 is a small GTP binding protein. It exists in inactive and active. It activated by GEF and inactivated by GAP. when it's activated to effectors, it increases vascular permeability. When you use the OFF6 specific inhibitor of this QS11, SECIN-H3, and now, then we can see the inhibition of vascular permeability, indicating that this pathway is required for specifically required for vascular permeability. So I'm not going to too much details about COVID because you are already know a lot of details about COVID. So, but just I want to highlight here is the COVID-19 not only damages lungs, but can also damage many organs. And one way it might be causing organ failure is through blood vessels leakage. So we want to know whether we can use the same pathway which proposed for bacterial sepsis for studying the COVID-19 mediated organ failure. So we just proposed here. So you have the COVID-19 SARS-CoV-2 virus here in the lungs. Probably when at high levels they enter into the bloodstream, which cause leaking of blood vessels and thereby organ dysfunction and death. So at cellular level, in COVID-19 patients, SARS-CoV-2 virus might bind to the ACE2 receptor through through the coat protein, thereby activate OFF6, which result in vascular leakage and thereby organ failure. So this is what we want to study. Unfortunately, we couldn't do that because of the lockdown of all institutes in the UK. So these are the people who done the work. Also, I mentioned at the beginning, and this is the place I work. As you can see, this is the seafront. This is the one of the two branches we have, which is a Singleton campus. The other one is called Bay campus. Okay, any questions?
Yeah. Thank you, Professor Venkat, for your excellent talk and giving a detailed explanation about the importance of GLP-1 receptor and the signaling pathways like G alpha, that is S and Q, and further on this R6 activation and vascular permeability increase, and which is related to the COVID-19 morbidity condition. So I have a question like we know this GLP-1 is like incretin like metabolic hormones where the
Sorry, I get it because it got stuck. Can you repeat the question? It got stuck. I couldn't hear your question, please.
Hello?
Sorry, I didn't hear your question because it's frozen. Hello, can you hear me?
Okay. Can you hear me now?
Yeah, I couldn't hear your question because it's frozen.
Yeah. Can you hear me now?
Yeah, I can hear you.
So we know that GLP-1 together with like GIP, these are incretin metabolic hormones which are identified. What kind of abnormalities we are seen in the patients, like diabetic patients, related to GLP-1 receptor?
You don't see any mutations, and there is a slight increase in GLP-1 levels, but because endogenous one is quite low, like its half-life is only 1.5 minutes, you never get the required insulin levels by endogenous one. So there are groups working on developing inhibitors for peptidase, dipeptidyl peptidase inhibitors, which are orally active. But I think it does cause a lot of side effects. Therefore, most of it, recent studies mostly concentrated on GLP-1 mimetics. So when you take more amount, you can see an increase in insulin levels to kind of supplement the demand required in type 2 diabetes, especially when you become insulin resistant. You need large amounts of insulin. Can you hear me? I think there is slight problem with.
I think the problem,
Yeah,
the connection problem with
I think some
Dr. Somu.
network issue.
Sorry,
I think maybe Logu, can you ask one or two questions to
We have
Venkat?
received
Yeah.
many questions from the participants. I will
Okay,
read out one of the questions.
Yes, yes.
Enlight the preferred expression system for the production of GLP-1, and also the enlight on the link between glycosylation patterns and immunogenicity response.
Okay, I can answer the first question, but glycogen pattern and immunity may not be related to my work, but I'll try to do. GLP-1 is produced from intestinal cells called L cells. Very often, GLP secretion is induced by food intake. When you take food, the glucose when passed through the intestine, which induce GLP-1 secretion. If you take glucose intravenously, then we don't see increase in GLP-1 production. So yeah, it is produced from the intestinal L cells.
Thank you, sir. The another question, are the localization of the receptors are stable? Could you set line on the localization factors?
Yeah, it is stable like many other G protein coupled receptors, but it is regulated by different aspects because sometimes GLP-1 receptor is more stable, whereas interleukin 13 receptor alpha 2, it is controlled through many regulatory proteins. And that's why we have to look for the cell surface expression when we want to target using a peptide-based drug.
Thank you, sir. The next question is GLP-1 are used in early diagnostic setting of type 2 diabetes, and which isoform is clinically validated as of now?
As far as I know, there is only one isoform for GLP-1 receptor, alpha 2. I think there are two types. One is called, the GLP-1 receptor alpha is only one isoform, which generally you can use it, but it's mainly expressed in very specifically in pancreatic tissues, so it's not possible to use for diagnosis purpose unless you get a biopsy.
Okay, sir.
But it can be used for cancer. People are using currently for pancreatic cancer. You can measure it in immunohistochemistry, and one way to do studies, you can use a radioactive compound tagged GLP-1 to localize cancer for diagnosis purpose by MRI.
Yeah, thank you, sir. Another question is related to COVID-19. Is it possible for heparin to target SARS-CoV-2 spike glycoprotein just like it was SARS-CoV?
It can be either you can use a antibody to neutralize it, but we have to be careful because sometimes spike protein is a very big molecule. If you are using a monoclonal, it might be masking only a small region of the spike protein that may not interfere with the spike protein binding to the receptor. Therefore, we have to clearly analyze which type of antibodies can act as can neutralize the binding. Another way I've seen people using is you can use a soluble form of ACE2 receptor, angiotensin converting enzyme 2 receptor. If you use a soluble form, what it does is it prevents binding of virus to the receptor on the membrane, at the membrane.
Yes, sir.
So you can make a small peptide.
Thank you, sir. Another question, based on the mutation studies, can you suggest some amino acids in GLP-1 which can be used for enhancing its potency and stability?
Yeah, it's been done a lot of work because we are working on the receptor. I have not discussed, but there's a lot of work has been done to change. In fact, most of them increase stability by changing the amino acids so that they no longer become substrate for this peptidase. For example, if you take exenatide, which is a natural exists in the nature because it's isolated from lizard saliva, but it is only have a 53% amino acids identity with native GLP-1, and it is resistant to peptidase breakdown. So already people have done a lot of work to prevent them to be broken down by peptidase. So those mutations already been done.
Yes. Now, another question is there any link between PD1 and IL-18 or alpha-2?
As far as I know, there is no connection because the GLP-1 receptor has a very specific functions in pancreas, whereas alpha-2 is a common one which overexpressed by many cancers, which is one of the so-called testis antigen. I don't know if anybody in the cancer know there are some specific antigens which express in males in testes, but over the time they become a foreign antigen for our own body, and therefore you can use those to develop antibodies for cancer treatment. Due to some odd reason, cancer cells express these testis antigens, and IL-13 receptor alpha-2 is called TAS or TA19.
Okay, sir. Thank you very much, sir, for answering many of the questions. We have lot of questions. We will share with you later. Thank you, sir. Formally closing the session. On behalf of organizing committee, we would like to thank Professor Venkat for giving an excellent talk on new treatment options for cancer, diabetes, and COVID-19. And also, he enlightened us with a detailed work on understanding the role of GLP-1 in diabetes, and also lytic peptidic targeting of breast cancers, and therapeutic targeting of sepsis and related to the COVID-19 research work. Thank you, sir. And also, we want to thank Dr. Somasundaram for sharing this session. We thank all the participants. Around 750 participants attended this particular session. Thank you all the participants. The participants are requested to join the next session at 5:30 p.m. using the same link. Thank you all. Thank you all.
Okay, thank you.
We shall be closing the session.
Okay.
Thank you, sir. Thank you.
Thank you, Venkat. Thank you so much. See you soon.
Okay, thank you.